This desire to categorize living things is almost as ancient as science itself. Aristotle was perhaps the first to codify a system for classifying animals by their internal and external similarities. During the mid-1700s, the Swedish botanist Carolus Linnaeus developed a standard method for naming organisms. He introduced the idea of genus and species. To him, animal organisms were perfect and unchangeable creations. 100 years later, Charles Darwin shook the world. His theory said that species could actually change over time. What's more, it also implied that all animals may have descended from a single common ancestor. Darwin suspected, if only we could travel millions and millions of years back in time, that we could witness the birth of the Earth's first animal. In the beginning, the Earth was devoid of living things. Then, about 3.5 billion years ago, something completely new appeared: life. For 2.5 billion years, single-celled organisms dominated the Earth. They were minuscule creatures, completely enclosed in a fragile membrane. These were living, pulsing beings, but they were not animals. Somehow, cells developed a language that allowed them to work together. When they did, it was a turning point for life on Earth. The very first animal had begun to take shape. But what was this first creature?
The first animal would really have been something very simple. It would not have had a head, a brain, a mouth, or any internal organs, not even bones. It would have been something made up of a bunch of cells, a bunch of animal cells. Those cells would have to cooperate, to work together for the common good of the animal. So what would such a creature have looked like? And even more importantly, is there anything like it still living today?
Incredibly, such an animal still exists and it thrives in these waters. And that is why Diaz has come to Indonesia. Sponges don't give up their secrets easily. Their apparent simplicity is both provocative and perplexing. At a distance, they seem solid. But a closer look reveals a labyrinth of tiny holes, tunnels, and chambers. Unlike our bodies, sponges have no definitive shape. They are as varied as their many habitats. Scientists have described more than 9,000 different species of sponge. They thrive in salt and freshwater environments around the world, turning up in the most bizarre and unexpected places, from the frigid waters of the poles to the warm seas of the tropics. Sponge cells are unique in the animal kingdom, but in order to study their miraculous abilities, Diaz and Erdmann will have to gather living samples and transport them to the lab.
We need sponges to study in the lab, and so we have to collect specimens. But we need to just cut a small section to be examined, and because it doesn't have a nervous system, we are not really hurting it. In time, the cut area will grow again as if nothing had happened to it.
All animals rely on cell-to-cell communication. But Diaz and others have been able to show that sponge cells have a unique way of working together. To witness these abilities is to be amazed.
Cell-to-cell communication is very important for all organisms. But in sponges, it's uniquely important because in sponges, cells carry out all the functions that other organisms are taken care of by specific organs, like the heart, or the nervous system, or the skeletal system, or the blood. So sponges have many responsibilities to perform.
Unlike most of our cells, those of sponges retain a special freedom. They can continuously reinvent themselves, performing some truly miraculous feats. If a sponge is passed through a sieve, its cells are separated. But somehow the cells still recognize each other. Over several hours, they begin to come together, building many new sponges. No other plant or animal can resurrect itself this way.
Most people, when they think of a sponge, they think on their bath sponge. And when you have a bath sponge in your hand, what you're holding is a soft skeleton that is made of collagen.
But unlike the bath variety, many sponges aren't soft at all. In addition to the soft collagen of the bath sponge, many have millions of brittle crystalline spicules embedded in their bodies. Spicules are microscopic structures that help give a sponge its rigidity and form.
Spicules are an incredible tool for us today to identify most of the sponges that are living in the oceans. You can identify a species or a group of sponges not only by the type and the size of the spicules they have, but the way that they are arranged in the sponge body.
For most sponges, the glassy spicules act as a kind of fingerprint. Magnified, their dazzling array of shapes and sizes are as beautiful as they are unearthly. Drifting along with countless single-celled organisms, we feel the tug of a current. In the wonderland within, this sponge's cells all work for the common good, but they do it in wildly different ways. As the current draws us on, we pass sponge cells that are making new spicules. The walls themselves are treacherous. They can simply engulf their unfortunate prey. As we are drawn through ever narrower passages, the current slows until we burst into one of the sponge's millions of tiny beating hearts. These are colonocytes, cells whose whip-like flagellae powers the pump. The flow quickens again as it whisks us away through the labyrinth of canals that lead out into the sponge's central cavity. What once seemed barely alive is actually a living animal, more complex than we could have imagined.